Hello, everybody, and welcome to episode number 82 of the Hyperthesis podcast. Thank you very much for joining us. We have an exciting episode up for you today. I'm Patrick.
I'm Pheely.
And I'm Liam.
Okay, so we are getting a little bit out of our comfort zone today with some new and interesting topics, especially for our main topic that Liam will be discussing about. But before we get into there, first of all, I want to apologize for not uploading last week. We tend to upload on a weekly schedule, but sometimes we're busy and it doesn't always work out. So we're going to make sure that we keep posting on the Instagram, and you can figure out how to find us later on in the episode if you aren't following us already on Instagram. So before we dive deep into the main topic, does anyone have an intro topic that they would like to discuss?
Yes. And sometimes life gets in the way, we have maybe sometimes valid excuse. You gotta pick up my daughter from school and such.
I was partying in Nova Scotia. That's my excuse. No, I was visiting my family, and I went and visited our old university and our old physics professor, and it was a good time. Got to talk about physics, so I think that's a justified excuse. This one time though, never again.
Yeah, never again.
Yeah.
We will not miss another one without announcing a break. Or we will.
My fingers are crossed right now. Just in case.
All right. So today's intro topic is something I found recently, but I think this is the discussion of a phenomenon that people are quite used to. Well, we all have, hopefully, at some point in our life, drunk the carbonated drinks, right? And this paper or this article that talk about this paper is talking about the effect of bubbling in these carbonated drinks that can be modeled. And the title is Dancing Raisins. A simple kitchen experiment reveals how objects can extract energy from the environment. So basically, you can make raisins, which are a type of, you call fruits, right? Yeah, these little piece of fruits that are really wrinkly, a lot of wrinkles, and you can make them basically float up and down in carbonated drinks. And that happens when you actually take a look at it. When you do it, you can do it with your, let's say, a clear beverage that's carbonated. It can be just like a soda. And when you have something that's wrinkly, bubbles can form easier, and those bubbles can attach to the object. And since the bubbles, they have density that's lower than water, which is because they are pockets of air, and they lift those objects, in this case, raisins, up to the surface. And then when it goes near the surface, the bubble popped, and the raisin will come down again. So there's kind of this back and forth of going more like up and down motion of raisins. So it appear to look like dancing raisins. So the actual phenomenon can actually be modeled and studied, because it's, in a sense, almost feel like a little bit of like basic buoyancy, right? Because the overall density of air bubbles and raisin has to be lower than the density of the fluid or the liquid is in, so it start to float. And the one thing is that the bubbles are kind of hard to create too, in like normal conditions. So you don't see that many bubbles form when you leave the drink alone, right? There might be some little bit of bubbles, but having raisins or anything, it be cloth fiber or things that are around, help create this, we call the nucleation sites, where the bubbles can form better. That's why, for example, when you pour beer, they say, oh, till your glass, but when you actually turn your glass back into like vertical, you create this air pockets when you pour that, which will create nucleation sites, so it forms more.
I imagine that the wrinkliness of the raisin allows for extra little bubbles to kind of get locked in against the raisin. Like if you had a grape, a grape in this beverage, most of the bubbles would be on like the bottom of the grape, I would imagine, right? And the ones on the top might float up. They might not stick to the grape quite as well as they would on the bottom, but because the raisin has all these little crevices, the bubbles might get stuck in there a bit more. I
Well,
don't know.
it's about the surface tension. So the bubble, to form a bubble, it has to overcome the viscosity, right, or the surface tension of the fluid, so the bubble can form. Which the raisin with its crevices and stuff help basically lower the surface tension.
Yeah, because the bubbles they have surface tension trying to keep them to the raisin, and then they're fighting buoyancy, and then so they have to kind of balance them out.
So, two interesting things about the study. One, it was published in Nature Communications, which, I mean, from kind of like the SciComm article that links to this study, that's just pretty funny, the difference, because the actual title is Levitation and Dynamics of Bodies in Supersaturated Fluids.
Yeah, I was gonna comment on that, but I decided to hold my tongue. I was gonna say how the heck are these things getting published in Nature and my stuff isn't? What the hell?
It's really interesting though because it's looking at these supersaturated fluids, and of course it's a Nature paper, so it's not mentioning raisins specifically, but it's looking at like the dynamics of that system in specifically supersaturated fluids, which is very interesting. Like, and something that we can easily access with carbonated sodas, for example, which are supersaturated with a gas, which we don't tend to think about as much compared to like supersaturated fluids with like salts in them or sugars or things like that.
Yeah, and I was just joking that like this research is kind of silly, but if you've listened to this podcast, you know that these type of papers are some of my favorite. The ones where it's physics that you might see it every day. Although I don't know how many people are putting raisins in their carbonated beverages, but in principle, it's something you could see very easily. And we still don't fully understand it, so people are studying it, and it has these type of papers always tend to have interesting applications that you might not expect. So I really like this type of research.
Well, if you look at the lore, I mean, I'm using a video game language here. It's actually originated from Dancing Raisins. If you look at the author's, I think, viral Twitter post, when they first put it on the archive, because he explained that when he and his daughter were playing around in the kitchen, this comes up, and he got curious about it, and it ends up being a paper later on. So the motivation is actually coming from the kitchen. Even though when you try to publish, you gotta make the title and the things more rigorous, right? And it's more generalized too, because it's not limited to raisin. It's any object that basically is similar topology.
We have a trend of talking about kitchen physics on this podcast for whatever reason. I think we have like a whole the physics of breakfast episode. So check that out. But anyway, back to the raisins, maybe.
Well, just looking at the experimental process, because this paper is really quite thorough in going through the experiments and the theories. So what they were able to do was essentially take this tank of water that was carbonated. They then depressurize and then measure bubble formation and the buoyancy. So what they actually used was not necessarily a grape, which is hard to control for because they have varied surfaces, very varied masses, and varied sizes. But they have a 3D printed sphere essentially that they're using, and they're able to measure the buoyancy of that by measuring changes in the weight within the glass of water. But the thing is that this 3D printed sphere is fixed. So they're actually measuring its buoyancy and how it changes based on it's fixed and not included in the scale. So they're able to measure these differences by just measuring the different weight or the different apparent weight of this glass of liquid, along with the loss of gas as well and how that changes the weight. So it's a really interesting. They have a very good picture here showing the 3D printed sphere at 0 minutes, 0.2 minutes, and 0.8 minutes, and looking at the bubble formation and just like how these bubbles are progressing on the surface of the sphere. So it's a really in-depth experimental process, and then it goes a bunch into the theory of what's happening, which is just really quite fascinating.
And also they do simulations, computer simulations too. Like the paper, if you actually read it, it's really thorough, quite comprehensive too. Maybe that's why it's in Nature, Liam.
Well, yeah, it has to be, right? They don't just take any papers in Nature. They take the ones who pay them a good amount of money. No, I'm kidding. The papers in Nature are usually pretty good, although I'm sure there's some stinkers in there. But so anyway, what did this paper tell us? What did we learn? What did the authors learn about putting raisins in carbonated beverages and modeling how they bob around and how the bubbles react and all that jazz?
One thing they were able to measure, which I think is really interesting, especially with these 3D printed spheres, and they were also able to simulate, was a mean rotation. So not only is there this kind of buoyancy that's changing as the gas bubbles form and then are released, but it's also causing these objects to rotate because the gas bubbles are adhering to the surface differently. So it's actually causing this net rotation. So in this case, they look at a sphere and using simulations using a discrete bubble model on a sphere. But I'm curious if you would see similar dynamics with non-spherical objects like grapes, where you might see this rotation happening because of a heterogeneous bubble formation on the surface.
Probably.
Yeah, the inhomogeneous nature of the topology, it's gonna spin differently and rotate differently and have different angular, like different moment of inertia, right? Like, well, so I think one thing I want to touch on is the supersaturation. I think this concept is kind of overlooked a bit in daily lives, but we see them a lot, right? Every pop can and pop bottles are what you say, I think, yeah, super saturated, right? Because they pressurized it. And at that pressure, at higher pressure, the carbon dioxide are more soluble in water. But once you turn the cap open or pop the can open, you are at atmospheric pressure, which are lower than where it should be. So it's less soluble. Then it wants to come out. The gas want to come out, but it's stuck in this solution. That's why when you put a finger in it, some people put their fingers in their beverage, or put some straw, like you shake it basically, or put some straw, stir it a bit. A lot of bubbles come out because imagine it's an unstable phase where it really wants to be gas, but it got stuck in this soluted form. What's called dissolved form. But by disturbing it a bit, really helps facilitate this nucleation and popping but the bubbles out. And this supersaturation you can see in dessert when you make syrup, I think. Because imagine you're trying to dissolve sugar in water, and you get to a point where it gets saturated. You can't dissolve anymore, right? But then it's still not very viscous. And what you do is that you heat it up. So when you heat it up, the sugar become more soluble. So the water can take more sugar in. However, when you cool it down, it doesn't just precipitate all the extra one that we just dissolved in. So they're basically got stuck in that dilute, dissolved form. And there's a limit to it. It will start precipitate at some point, but it allow us to dissolve more, what's called more material or whatever you want to dissolve in. And I think this technique used a lot in chemistry. And also, it's easier to dissolve things generically when it's hotter, right? When your solvent is hotter. So this effect of super or this phenomenon of supersaturation is really common, and I would say it's a little bit overlooked.
Definitely. I'm wondering if there's any, because at the end of these type of papers, usually at some point they'll have some list of applications that they'll say like, oh, this might have applications in industrial lines or in certain types of experiments or something like that. I'm wondering if this has applications anywhere, if you manage to see that in the paper.
Yeah, this one is actually maybe you could be able to apply it in geology or to study volcanic activity, because what happens in like volcanic eruption, is basically the same thing, the same phenomenon that we have here when popping the can, right? Because you think of the magma that's underground and it has a lot of pressure on it. So all these gas and things that would dissolve in these rocks were in it. Once it comes out to the surface, there's a lot of they got depressurized, and the gas and things that dissolve in it, maybe they want to come out and create this violent eruption. So maybe this process can be modeled through this mechanism.
Just reading the final paragraph of the paper, the final sentence, unless someone else was going to say this, it's, quote, future work exploring body shape, multi-body dynamics, and fluid structure interactions is likely to prove ... fruitful.
I
Nice
can't
little
I
pun
can't
there.
believe that. I can't believe they put that on a Nature paper. That's incredible.
Yeah, very well done. And well, thank you very much, Pheely, for bringing that thrilling dance of supersaturated bodies into our midst, and I guess extending the lore of food physics that we've discussed. But moving on to our main topic, going just completely switching directions. Our main topic today is focusing on something called quantum biology, and I will let Liam take it away as I know nothing on the subject.
Well, luckily for you, I don't either. I basically, yeah, I wanted to talk about this quantum biology field of physics, and I've heard about it over the years once or twice in a few different articles, and maybe you have too. You'll let me know by the end of it, but I haven't really seen too much of it in my career aside from these one or two articles. So maybe that's just because I was unlucky, and it's a and my field is so niche that I haven't really noticed it, so I'm not aware of it, or maybe it's because it's kind of a niche field. And I'm not a biologist or chemist by any means, so this kind of area of physics is very quantum biology. There's a lot of biology and chemistry in it, so it's not really up my alley either. So that's why I kind of wanted to look into it. I wanted to read up on it a bit, just so I was aware of what is under the scope of quantum biology. So quantum biology is it's kind of what you might expect it to be. It's the study of biological mechanisms and phenomena that cannot be accurately described with the classical laws of physics or chemistry. So just like quantum mechanics, sometimes classical physics fails, and you on very small scales. So that's where quantum mechanics comes in, and that's the best theory we have on those scales. Turns out that there's a few situations in kind of biology and biochemistry where classical chemistry and physics doesn't really work. So people tried to use quantum mechanics on these small scales to kind of remedy that. And currently, according to the internet, which is where I found all this information, uh, there's about right now there's four major life processes that are influenced by quantum mechanics that are not fully classical. And some of these people may have they're certain about some of them, and some of them are more controversial, and they're just theories so far. So these four life processes, they are how certain enzyme they're not fully classical and slightly quantum, we think. Certain enzyme catalysts is the first one. Certain sensory processes are predicted to be slightly quantum. Information encoding, which I'll come to what that is in a bit, and certain energy transfer processes. So, like photosynthesis, for example, which I think out of all four of these, the energy transfer one, I'm probably not going to get into very much for the sake of time, but it's arguably pretty interesting. So, that might be a talk for a future episode. Maybe I can go into the details of how people think quantum mechanics appears in photosynthesis. But for now, um, I'll kind of go through these four life processes and talk about how maybe quantum mechanics comes into play. So the first one was the enzyme catalyst. So enzymes are these proteins that act as biological catalysts by accelerating chemical reactions within a living organism. And they've actually been postulated to use quantum tunneling to transfer electrons in these so-called electron transport chains. So essentially it's possible that some protein structures have actually adapted over time to enable sustained conditions for quantum tunneling in biological entities. So just, I guess, quickly, I'll get into that in a bit, but quickly, what is tunneling? Quantum tunneling is this phenomenon where if you have a particle that's very small, so it's a quantum particle, it may suddenly pass through a barrier that's higher energy than the particle due to this kind of wave-particle duality that occurs in quantum mechanics. So you always hear this analogy is that, if you run at a wall, you're going to hit the wall. But according to quantum mechanics, there's a small probability you'll just phase through the wall suddenly. Now, that's a bad analogy because, I can run at a wall a million times and I'm almost 100% certain I'm not going to pass through it. But on very tiny scales, if you're a very small person and you're running at a very small wall, although the wall has to be bigger than you, classically you won't pass through it, but quantum mechanically you can. So that's kind of my hand-waving explanation of quantum tunneling, and it's real. We've measured it. It's used in kind of electronic. There's actually, I think there's quantum tunneling diodes in electronics, and anyway, we know it's real. It's not science fiction, and it turns out that in these kind of enzyme catalyst processes inside living organisms, it might occur.
Well, when you say it might occur, like the one thing about quantum tunneling, I think is like it's very small, right? Like the effects are quite small, and you look at the scales of the enzymes, they're quite large. Like at molecules, when you do in school, when you do like quantum tunneling stuff, it's always like, oh yeah, do a square well, finite square well, and it feels like the level of the magnitude of the complication is much higher when you have molecules and enzymes.
Yeah, and that's usually why you don't really need quantum mechanics when you're talking about, human cells or plant cells or even very large molecules. You can typically ignore it. So what scale are we going to talk about here? Is quantum tunneling really feasible? So I'm trying to figure out how to best explain this, but basically, in the organelles of cells. So organelles are like the little organs inside of cells. So like your nucleus, your mitochondria, your things like that, which I completely forget since high school what they do and how many there are, and I have vague memories of this. Um, but these organelles, what happens is that they transfer energy through hydrogen ions. So they use hydrogen ions to transfer and process energy, and it turns out that at little proton distribution sites within the organelles, and I don't know exactly how this works, and I don't know which organelles have these. I apologize, but in these organelles, there's these little proton transfer sites, and it turns out that the distances where this stuff occurs over is on the scale of an angstrom. So that's a scale where quantum mechanics becomes relevant, because an angstrom is kind of the diameter of a hydrogen atom, roughly. And a hydrogen atom, you can't fully describe it without quantum mechanics. So to experimentally measure how quantum tunneling can come into this, you need to look at these proton transfer chains somehow. And I think I looked at a couple papers and they were way over my head. But it seems that there's one thing I did recognize, and it's that from a physics point of view, when things are small but not super small, so an angstrom's pretty small, but it's big enough that you can use semi-classical approximations, which is something I know a decent amount about.
Uh, just to add in, just for these organelles, just to add a bit of biology into it. So the mitochondria is a place where these processes can happen because you have an electron and a proton exchange. And essentially, there is an electron transfer reaction that occurs between a membrane within the mitochondria. And that transfer process essentially stores a gradient of protons. So there's more protons on one side of the membrane than the other. And those protons are only able to go through certain channels to cross the membrane. And those channels utilize that potential energy being converted into chemical energy to produce adenosine triphosphate, which is what we use to power a lot of the functions within other organelles within our cells. So it's ATP. So this proton transfer mechanism is used to produce ATP. And so I believe that this is where it's happening within those specific enzymes that are able to convert essentially potential energy from the protons into actual, use that energy to produce ATP.
And somehow, somewhere in there, you're getting some tunneling through some potential such that the kind of transfer of energy, the transfer to chemical energy occurs. And I read up a little bit on it, and it was just kind of, I don't know, it was beyond me, but it's good. It was interesting to see that this kind of stuff is actually being kind of considered. And some of the methods they use were these semi-classical methods. Yeah, because you have these kind of biochemicals which are macroscopic, so they're fairly classical, but you have things happening within them on the scale of an angstrom, which is a kind of a quantum scale. And semi-classical methods are way simpler than purely quantum mechanical methods typically. So for the physicists out there, you can take Planck's constant, which is this constant that shows up in quantum mechanics, and you can assume it's really small, and then you can make all kinds of approximations with it. You can, when you have small and large parameters in physics, you can do lots of theoretically fun stuff instead of just trying to exactly solve a problem that might be very hard, which is actually something Pheely and I did in a paper at one point, although nothing to do with the mitochondria.
Well, I'm thinking of another application of tunneling. Quantum tunneling is the scanning atomic microscopy, right? Where it's used the opposite effect because the same effect but inversely. So instead of trying to send something and tunnel through, we try to receive what's tunneling through us. So what happens is that atoms, if you imagine as the sphere, right, and has nucleus in the middle and clouds of electron around it, like a sphere. It might not be a sphere, who knows, right? But actually, we kind of know this is shaped close to a sphere. So what happened is that, so if you have electrically charged tip, that allows the current to flow through, and it's this really small tip that you want to use like a needle, try to scan the surface, what the shape look like. When it got close to the boundary, the edge of the atom by near the electrons, because the tip are so thin, the electron would tunnel through a little bit into the tip, right, and create this very small current, but that you can measure. So and it depends on basically how close you are to the atoms and what charge is like there. You can scan it by moving it all around the surface and create this, instead of like depth map of the ocean, but it's basically the electronic map that represents the shape of the structures, or like on the 2D plane of the structures. You can see like, oh, maybe atoms, these molecules look like this, or these atoms look like this, arranged kind of like this in this 2D plane.
Are those the are those I think I've seen images of those where they might have like, a benzene molecule or something, and they'll try it looks like a bunch of little mountains of probability. They kind of map it out like that. Would they use it to make something like that maybe?
I'm not sure about that, but
Okay.
one of the thing that they use, because like quantum tunneling, one thing that really it requires is a thin barrier. Because if it's thick barrier, it would just reflect off it, right, or get absorbed. There are two ways. Classically, when the wave comes in, it's either get reflected or absorbed. But what requirement is has to be thin enough. I'm not sure if the enzymes thing you were talking about, like, what's the magnitude or like the barrier that's trying to tunnel through. Because one of the problem is that a lot of things, the microscopic structures we have, they're really thick, and quantum effects, unless the wave is really, maybe is it high in energy, has to be high enough to have the quantum effect to be able to tunnel through it too. They probably can do some calculation on the requirement for that too.
Yeah, it's all quantum mechanics. It's all statistical, right? Which we talked about in an episode recently a little bit. So it's one of these things where maybe these are quite high energy. So even if if the scales, if I understood what I read correctly, if the scales where this is happening is 1 angstrom, that's pretty big for tunneling, I'd say. I mean, I don't know. I need to learn more about kind of the experimental side of tunneling. There's a group near where I am, led by the physicist Ephraim Steinberg, and my supervisor talks to them a lot, and they're very interested in calculating tunneling times. And when you excite an atom, how long does it take for the electron to hop down to the next level while it emits the photon? Because according to our current theories, it's like pretty much instant. But surely it must have some time scale, right? So there's a lot of people kind of researching this stuff, which is very interesting. But if things are happening on the scale of an atom inside these kind of enzymes, or inside these organelles, what might be happening is just tunneling could be happening if the um the enzymes or the electrons are very high energy, or if you have a lot of electrons going at, it's like if I have thousands and thousands of electrons hitting a barrier, even if there's a very low probability for them to pass through, the more of them there is, the more they're going to pass through. So maybe that's happening.
So there's actually a paper called Medical Hypothesis: Neurodegenerative Diseases Arise from Oxidative Damage to Electron Tunneling Proteins in Mitochondria, by James P. Bennett. And what this goes into is talking about how there are enzymes specifically geared towards this electron tunneling process to lower the activation energy, which is the whole point of enzymes. But the fact that these enzymes getting oxidized, which happens with a lot of things in our body because we need oxygen, it actually leads to degenerative diseases such as ALS or Alzheimer's, Parkinson's. So it's just a hypothesis. So it's like just a medical hypothesis, but it's very interesting that's able to kind of relate that to more macroscopic degenerative diseases that we have.
And that's what I find interesting about this quantum biology stuff, is that some of it's very much, like you can argue, well, atoms exist because quantum mechanics and we're made up out of atoms, so quantum biology, voila. But I'm more interested in like the tunneling and the entanglement and the kind of like the fun quantum buzzwords where they come in, and those tend to be a bit more controversial. So a lot of this stuff has been theorized and it kind of makes sense, but some of the experiments have mixed results. So they could be true, it might not be true, or maybe we're missing a key piece of it.
So aside from tunneling, what other quantum processes happen in the body?
Well, you mentioned vision. So the second life process where these quantum mechanical effects might occur are certain sensory processes. So vision, for example, I mean, again, this isn't the kind of buzzword stuff I was just talking about, but Vision relies on quantized energy in order to convert light signals into these action potentials that send signals to your brain. This is a process known as phototransduction. Fun fact, the human eye can actually, or the retina, I guess, can actually detect single photons if they're high enough energy, and if they're not high enough energy, it can't. Although in practice, that's a bit hard to do. If you try and put yourself in a very dark room and you fire one photon into your eyeball, it turns out that there's a lot of like false signals that your brain creates. So it's hard to kind of separate this one photon from all that background noise. But it has been done, I think. Maybe people who are very calmly minded don't have as much background noise firing.
Well, it also, remember our episode with Austin, right? Vision is more than just electrical signals. So it's how our brain interpret that, and also,
Mhm.
it got filled in a lot. Our brain do a lot of background work that we don't realize consciously. So I think even though it's technically one photon can trigger that mechanism, it doesn't mean it's a part of vision. You know what I mean?
Yeah,
It's
yeah.
different from, let's say, the photo camera, right? The sensor, maybe it's that sensitive, but then you got a signal and then you decide how you process that signal. However, and our vision, our eyes, I think, is that, well, even though it might create a signal, but the processing of that signal through the brain, that's a mystery that I think we don't have. We have, I think, pretty okay understanding, a pretty decent understanding on how that comes about. But I think that's from learning through, not through building fundamental blocks, exactly how like each cell processes a signal, but learning heuristically, or at learning from evidence and from experiments and what we see, right? So I don't think we have like a picture perfect understanding of mechanism of translation from or electrical signal that comes from the retina to the vision that we see.
Yeah, well, I think you're right, because so that's why I wanted to bring up vision. So when light, this phototransduction, when a photon interacts with the light receptor in your eye, so your retina, and maybe I don't quite know how retina work, but the idea is that the receptor absorbs the photon and it changes its structure, and that results in a visual signal through this action potential. So it sends a signal to your brain, but it turns out that this process occurs at a very rapid rate. So I don't know how long it takes a signal to go to your brain, but I think the fact that receiving the photon and creating the signal and then sending it, it happens in under 200 femtoseconds, which is a very short amount of time. And many people suspect that because of this very small time scale, quantum effects quantum effects are happening in there. And the article I was reading mentions quantum effects somehow are in play when shaping the ground and excited states of these action potentials in order to achieve this very fast efficiency. But I was not able to look into that much more. So more research needs to be done on my end at the very least.
Well, I think from what you said, if it's that fast, well, why don't our eyes have higher frame rate? Is the limitation on speed? Is it the processing speed of our brain that is a limiting factor then? Because if the signal can be processed, I mean, if the retina or the cells can process in like 200 femtoseconds and give you a signal, there must be a signal at a frequency of the period of about 200 femtoseconds, which is definitely more than 30 FPS, right?
Definitely.
Or like more than 60 to more than 100 FPS for sure, like much more. But um, an average human being will still struggle to, let's say, you move your hand from your face really quickly. It's not like you're missing certain parts, right? It's not like a smooth glide. So there's limitations. But from what I hear from you is that, well, if the process of converting light into electrical signal or this action potential can happen at these very fast, very low period. Does that mean the actual processing that our brain does have this delay that comes into the our frame rate of our eyes, basically? Because what people do in the video processing, especially real-time video processing, you want to have your frame time or the time you use to render or process the frame really low so you can have higher frame rates, right? And that's the limiting factor.
Yeah, I don't have an answer, but I'm gonna guess that something like you said is probably the answer. It's if I take a page out of the particle physicist book at CERN, they get a crap load of data. They get so much data from their particle collisions, and then a bunch of it gets thrown away, and then they average out a bunch of it, and in the end, there's way less. So I'm gonna guess that if you're receiving all these signals every 200 femtoseconds, right? Your brain probably averages that out, and it probably throws a bunch of it away, and then it probably can only process it, so fast, and then what you get is 30 frames per second or something like that. Again, I have no qualification to say that. That would be my very naive guess, which maybe is partially true and maybe is completely wrong. I don't know. But interestingly, another sense that might relate to quantum mechanical effects is smell, olfaction. So, on a basic level, smell can be broken into kind of two parts: the detection of the given chemical, and then how the signal is sent and understood to your brain, just like vision, except now it's not with light, it's with little molecules that go up your nose and they smell. So, one theory, which I think is one of the more popular one, is called the shape theory of olfaction, which basically predicts that certain smell receptors are triggered by the shapes of certain chemicals or molecules. And whenever one of them's triggered, it sends a specific message to your brain. So that's why, when I smell an apple pie, it smells like an apple pie. It doesn't smell like Play-Doh or asphalt or some other unrelated substance. However, this other theory proposes that smell receptors actually detect the vibration of the chemical molecules, and certain smells are actually due to the different vibrational frequencies due to quantum mechanics. So this theory is called the vibration theory of olfaction, and I think it has something to do with the idea that these vibrating molecules, when they go in your nose, they hit a receptor, and only certain ones, there's actually, again, a tunneling thing happening where only certain molecules actually tunnel into these receptors, and other ones are not likely to, due to their certain frequencies, which I don't fully understand. But this theory was interestingly proposed in 1938, and then it had a bit of a resurgence in 1996. And there's a bit more to it, which I don't understand and can't do justice. And this theory is a bit more controversial too, because various experiments have been done for it, and some of them, certain animals are actually able to distinguish smells based on their different frequencies, even when they have the same shape, while other experiments show that animals could not distinguish smells due to the distinct molecular frequencies. So I didn't look into these studies. I don't know if these were all with the same animal or with different animals, or maybe some animals can and some can't. But it's kind of like, I don't know how you would ever come up with this kind of stuff. It's kind of interesting. So
Yeah, I mean, certainly animal experiments are very difficult to do, and even trying to figure out, for example, how people smell and how animals like dogs can smell is so hard to do because dogs can't communicate. They can't say, "Oh, that smells the same." And even though the signal looks the same, there might be very slight variations or ways in which it's activated that means that it will smell different in your brain. And there also, just a fun fact here is that the smell of sulfur or the smell of rotten eggs, it smells really bad, but it doesn't actually really have a smell to it. That's the nerve, like the receptors dying in your nose, because that smell of rotten eggs actually kills those receptors.
well, I was about to say something like Pheely said earlier with, maybe your brain is interpreting it a certain way, or maybe it's a false signal or something like that too. That could be happening. So that's interesting that the rotten eggs, you're actually just smelling your nerve, your those sites dying. they grow back, I guess, because I've smelled a lot of rotten eggs in my life, and I can still smell.
Also, you can get into like philosophy too, right? Or like
Oh no.
existential. That's like, well, I mean, like when you say the same smell, is it really the same smell? Because you could say the same thing with color. Someone see red, and we all know it's red, right? Like we all agree this color calls red. But it's the red we see. I see the same red you see. But we have this language that we agree that's red.
I think the way I think about that is maybe we all see slightly different shades of red, or maybe, we all, when we smell an apple pie, we all smell a slightly different thing. But to a good approximation, I'm going to say that probably pretty much the same. We're all born with pretty much the same DNA, and we all grow a certain way, and everyone's different, right? But I'm gonna my guess is to a good approximation. We all smell basically the same thing, but I guess when it comes to taste, for example, people don't like certain tastes, and some people do. Like I don't like cilantro, right? And most people are like, "What? You don't like cilantro?"
I was
So
gonna bring that up. It's
yeah,
like,
yeah,
if you
exactly,
don't
right.
like the God blessed plant, the best plant on the planet, I don't know, man. Maybe you just have inferior genes.
Yeah, probably. It tastes like stink bugs to me. So maybe that could happen with smell as well. Because like there's people, I know people who like certain smells and I don't like them. So I don't know. That is, yeah. I kind of shook my head when you said start to get into philosophy, but I'm kind of into it now.
I will just say there is a very good Vsauce video that was done many years ago on the topic of how we see color and how we perceive color. And then, of course, he gets very philosophical and big picture with it. But I would highly recommend checking that out.
So this is the next life process. So out of the 4 life processes I mentioned, this is the one that really made me kind of want to look into this topic, and you guys might have actually heard about it too. So this is the one. It's a third life process that it was the. Oh God, I forgot to write it down. It was the information one, information encoding life process. Which, what does that even mean? I'm not 100% sure, but one example of it is magnetoreception, which is a process where animals can detect the very small magnetic fields of the Earth in order to orient themselves and gain direction. So birds migrate; they'll follow magnetic fields. Even like some bugs, like certain cockroaches can detect it too.
Well, they say I don't know how true this is. That's like birds have little magnets in their nose, and humans used to have them. That's why we have little holes or something. Maybe there was some um science myth that I learned when I was in grade school that like, oh, these are cavities in our nose because we used to be able to detect Earth's magnetic field, blah blah blah. But we'd rather be smart than use our organs.
I'd rather be smart than use our organs. Incredible. I think
Oh,
I
isn't it
think
true? Isn't it true? Like,
yeah, yeah.
what are these claws for or nails? Oh yeah, we don't need them to slash thing or slice thing. We just build stuff to do it.
You use them to play your classical guitar. What do you mean?
Yeah, but to think about it, right? Like cats, for example, use claws to hunt. Humans, I think when we were like million years ago, we use our nails to fight. But now, we invented jujitsu and Muay Thai and martial arts and guns and weaponry. We use our brain so we don't have to use our organs. Think about it. We become, two-legged, right?
Yeah, yeah.
Or
No,
we
no.
are not using these arms to run.
Yeah, yeah. I agree with what you're saying for sure. So, but I think that birds having magnets in their nose thing is not. I don't think that's the case because we scientists don't fully understand how birds and other animals can actually how this magnetoreception works. And it was the first example, a potential resolution to that was the first example of quantum biology that I was made aware of a few years ago, and kind of what got me interested in the topic. And I wrote it down as a talk about it on the podcast later, and I'm finally here. So one possible explanation for magnetoreception actually uses quantum entanglement, and this is the one you guys may have heard of. I don't know, but it's called radical pair. It's called the radical pair mechanism, and it's this well-established thing in spin chemistry, which I don't fully, I'm not a spin chemist, whatever that means, but particles have spin. It's this quantum mechanical property. What is spin? It's kind of like angular momentum, but it's not. We know how it works pretty well, but we don't really know what it is. We've talked about it a few times before, but spin's important in quantum chemistry. And in 1978, this was kind of a resolution that was proposed to apply to magnetoreception of animals. So in quantum mechanics, you can have entangled quantum states called singlet or triplet states, which are these kind of superposition of spin states. They're quantumly entangled, and we've talked about entanglement a lot before, but the idea is that if you have two entangled particles, they can each be in one of two states, and they're very far away. If you do something to one of them, because they're entangled, something will happen to the other one, even if they're really, really far away. That's the idea, and we have experiments to kind of prove this, and there's a whole branch of physics, and you've talked about Bell inequalities before, and it's big in quantum information science, these spin states and Bell states. But anyway, these entangled quantum states, which are in superposition of spin states, the idea is that for animals, if they have some kind of particle or molecule within them that exists in these spin states, the spin states could potentially interact with the magnetic fields of Earth in a way which could create a signal and send it to the animal's brain, telling them what direction the magnetic field may be going. So in the 2000s, the molecule cryptochrome, which I think I'm pronouncing correctly, was proposed as a molecule which could actually possess these entangled spin states and actually create these kind of sensitive radical pairs. So cryptochrome, it's this protein that's found in the eyes of European robins and other animals, and it's the only known protein to form photo-induced radical pairs in animals. So basically what that means is that when light enters their eye, it kind of creates, it somehow creates this entanglement, and then the entangled particles interact with Earth's magnetic field, and then it tells the bird's brain where to go when they're migrating, for example. But we know that birds and other creatures don't need light necessarily to detect the magnetic fields. They can fly at night and they know where they're going. So it's not a complete picture, but it's a start at the very least. That's my understanding of it. And it also seems that this cryptochrome, it's only these radical pairs which cause these entangled states to do their thing, is only primarily triggered by blue light. So there is a lot of blue light in the sky, but at nighttime, maybe these birds' eyes are sensitive to very low levels of light, and they can still do it effectively at night. Maybe in the daytime it's more effective. I'm not sure, but there's experiments which support this basic entanglement theory.
I mean, I think the sense of direction has been lost, right, for us. Like we don't really know unless we look around, right? Like, are we facing north or south? But I thought there were some people in, like, it was either Australia or New Zealand, because it's a language, how it works. Like they don't really have left and right or something like that, but it's more like north, south, west, and I'm trying to recall, because maybe there was an idea that no, they actually can sense the Earth's magnetic field, but that might be a myth or a hoax. But the language actually shape how we think. Like I think in Japanese, the left and right is the opposite or something like that, or it's different from subject on object. Left and right is different.
Yeah, I can imagine that if you grew up with a language that didn't have the concept of left and right, and you only had kind of, north, east, southwest, you'd probably mentally keep track of where was north, for example, as you moved about your business. I'd be walking down the street, and in my mind, I'd know to this direction's north, and as I rotate, unless I'm doing any crazy somersaults or anything, you kind of mentally keep track of which direction's north, and then from that, east, south, west. So maybe that could be what was happening. I don't know.
Or when people play video games, right, and you have minimap, and sometimes you can choose for it to rotate with you. So you always point up, right? Or you can choose to have your labels or your directions on the minimap fixed, so north is always point up. And some people are more used to the other, right? Like each one is like personal preference, but I think it's have a deep implication of how our brain process information and direction.
Mm-hmm. Exactly. So maybe something like that's happening. But just to kind of wrap this up, there's these experiments which support this basic entanglement idea, and that they show that this process can be significantly influenced by these very weak electromagnetic fields of the Earth, and even um, the direction of the magnetic fields of Earth has effect. So if you take these specific kind of molecules and you these spin states and you change the direction of the cryptochrome and stuff like that, they can detect that. So it's good evidence that maybe this is actually what allows birds and other animals to do this. But we still don't fully know if this is truly the case or not. This is still not a completely resolved question. Researchers have found evidence for this entanglement as the mechanism as magnetoreception within European robins and cockroaches and garden warblers, which I'm not 100% sure what those are, but it's some kind of bird, I think. And they could no longer, what they did was they actually exposed these creatures, these animals, to a specific radio frequency which kind of obstructed the magnetic fields of the Earth, and they could no longer find their direction. But I guess that's to be expected because they use the magnetic field to steer themselves. So if you obstruct it, of course, they're not going to know where they're going. But I think understanding this is important because these specific radio frequencies, they can study if they interact with this cryptochrome in a specific way and if they affect the spin states as well. And there's some other reasons why this could be the case and why this might not be the case. For example, they studied the cryptochromes in chickens and pigeons and found that they were way less sensitive. There's actually different kinds of cryptochrome, and in chickens and pigeons, which are non-migrating birds, they were way less sensitive to magnetic fields than they were in the European robins, which are migrating birds, which kind of suggests an evolutionary optimization of this protein as a sensor of magnetic fields. So there's evidence for it. It'd be very cool if that was the case. It'd be nice to people ask you about, quantum entanglement. You're like, oh, well, birds use it. That'd be kind of cool. But, well, we still don't know yet.
Well, with that mystery lingering over, we'll move on to how you can reach us. And I will just say, Liam, do you have any final words on that?
I said there's 4 life processes, and I only gave 3. But the 4th one I'm not going to get into. As I said at the beginning, it's how quantum mechanics has effects of energy transfer in living organisms. So like photosynthesis or cellular respiration or things like that. So that's arguably a very interesting topic, how quantum mechanics comes into play in these processes in living organisms. But for the sake of time, that's not a topic for today. And I think maybe in a future episode, I'll try and go through at least photosynthesis. But we'll see, because I'd be interested to see how quantum mechanics comes into play there, and that maybe it helps solve the mystery of why trees are green, for example, or probably not, but who knows?
Who knows indeed. So if you're an expert in photosynthesis or looking at the quantum aspects of biology, feel free to get into contact with us. Also, I will say, if you're interested in learning more about the magnetic frequencies and everything that happens with magnetics on Earth, you can go listen to our episode with Zoe Vestrum, where we looked at magnetotellurics and how we can measure things inside the Earth using magnets. If you are interested in contacting us, no matter what you study, or if you have questions, comments, concerns, you can reach us over email. We are hyperthesispodcast@gmail.com. You can also find us on Instagram. We are at the Hyperthesis, where we will post updates, especially if we miss episodes, which we said we wouldn't be missing again. Even though Liam crossed his fingers, we'll be holding ourselves accountable for that. Although we will be probably going for a summer break quite soon. So we will keep you updated with all of that on our Instagram. Now, if you're listening to us, congratulations, you did find us. We are based out of Spotify, but we can be found on pretty much whatever podcasting service you use, whether it's Apple Podcast, YouTube Music, Audible, Wherever you get your podcasts, you can find us. So make sure to share us, give us a rating, subscribe to us, especially on YouTube, because we do have a YouTube channel. We are up to date with our YouTube videos. You can check out the wonderful animations that Pheely has done, and which take a long time to process. So try and watch those YouTube videos, and who knows, maybe one day we can pay for the electricity that's used to process those videos. In the meantime, though, give us a rating, share us, subscribe, tell your friends, and if you are an expert in your field, we would love to hear from you and have you on the show. So, moving on to the final story, we'll make it a little bit quick, but I do just want to go over, since we've been talking about biology so much and different biological processes, I think it'd be fun to go over a brief history of taxonomy. Now, taxonomy itself is the study of how the tree of life is arranged. So taxonomy arrives originally from the ancient Greek word taxis, which means arrangement, and nomia, method. And so therefore it means the scientific study of naming. And now in biology, it's used as the scientific study of classification and looking at the relationships that developed and have developed as evolution has proceeded between different organisms. Now, there's a taxonomic hierarchy in biology, which starts at the very top using domain, kingdom, and phylum, followed by the class, order, family, genus, and species. And we tend to use, for referring to a lot of different species, just the genus and the species. So for example, humans are Homo sapiens. And more specifically, if you want to get into the subcategories of species, we are Homo sapiens sapiens. Now, we didn't always classify things like this, and humans, of course, do love classification systems and putting things into groups, but it hasn't always been as straightforward, and even today it's not as straightforward.
I think I'll expand a bit on your etymology, because I think nomi is usually from the word nomos, which means convention, right? So it's not something that like logos, for example, logi, which is like knowledge that's that's something for another day. But nomos is something like language, for example. It's a convention we decide on. So taxonomy is a type of classification that humans decide.
Yes, yeah. I mean, we can. I think it'd be fun to do an episode on name origins, especially since we use so much in the different sciences. So looking at the roots of the different words would be cool. But going back to taxonomy, there's really not an extensive history of taxonomy. Of course, if we go back as far as the ancient Greeks, including Aristotle especially, he worked on things that might resemble taxonomy, but we really didn't see a lot of movement in the field until the 1700s. So really trying to distinguish and name and categorize the different types of plants is important, of course, along with the other animals. So it's important to recognize, is this plant poisonous or not? How can we identify it? How does it relate to other plants? And how can we make sure that we can pass on this knowledge so if someone eats a poisonous berry, it's not being eaten by other people. So we can create this kind of warning system that's a preliminary classification system. So, of course, Aristotle, he did some work related to this. So, and even further back, the ancient Egyptians, they did work. But of course, we have decent records from Aristotle, and he began to classify living beings by their parts or different attributes that they have. So, for example, how many legs, if they lay eggs, are they warm-blooded, are they cold-blooded? And really, he divided all living things into two groups, plants and animals. Now, of course, some were very iffy, and I think it's worth skipping this part in time because we did try and group things, especially with Aristotle and some of his students, but it wasn't really effective. It wasn't something that, well, it is something that held on for a long time, but its effectiveness was not that great. Because these Aristotelian systems, of which there were many, were used throughout the Middle Ages as well. So there wasn't really a lot of advancements. Of course, there was more discovery of creatures, but we didn't really see any great changes to the system that Aristotle introduced. Now, there were certain concepts like the great chain of being that was introduced into this kind of Western pre-academic thinking, but this was ultimately derived from Aristotle's idea. So, unfortunately, Aristotle, or fortunately, his ideas stuck around for quite some time. And even during the Renaissance, the categorization of organisms became more prevalent, but there was no really great standard. Now, some of the earlier people in the kind of Renaissance or the middle of the 1000s, we'll say, so the 1500s, 1600s, there was some work, including work by someone who's been called, quote, the first taxonomist, and this was an Italian physician by the name of Andrea Cesalpino. So he was actually able to write a book called De Plantis, which came out in 1583 and described more than 1,500 different plant species. And he was able to recognize 2 large plant families within this book. Now, there wasn't much movement from this book, which came out in the late 1500s, until the mid 1600s, with the works of John Ray, who was English. Now, he was able to make what was known as the Methodus Planetarum Nova, which was published in 1682, which detailed over 18,000 plant species. And at this time, his classifications were the most complex, done by any taxonomist that had been published, and he based his taxa, again, on characteristics, much like Aristotle and much like Cesalpino. Now, the next major work was produced by Joseph Péton de Tonnefort, who in France published his work in 1700, which included 9,000 species in 698 different genuses. And this work actually directly influenced Linnaeus. So Carl Linnaeus was a Swedish botanist who is pretty much the father of modern botany and taxonomy. So his major work, which was called the Systema Naturae, which the first edition came out in 1735, followed by his Species Planetarum in 1753. And he came out with the 10th edition of that a few years later. But Linnaeus was actually able to revolutionize taxonomy by coming up with more advanced ways in which we can group organisms based on their characteristics. So he used a standardized binomial naming system for plants and animals, which really proved to be elegant and tamed the chaos from these previous systems. So it made it easier to identify plants and animals and work down through a tree of sorts in which you can start to identify more specific parts. So, for example, this is a tree. Okay, this tree has broadleaves. Okay, this broadleaf tree, the leaves are this shape. And so he really developed this system in which we can start to classify things easier and group them based on similarities and differences in certain characteristics. So the Linnaean system is something still used today, but of course it's been advanced quite a bit with a lot of modern technology. So we have a whole field of bioinformatics and also DNA sequencing, which we've talked about before, which is able to better identify differences in species at the gene level. So, and we are also discovering many, many more species. So instead of looking at characteristics that are physical, we're looking at molecular characteristics in their DNA. And of course, I could talk about DNA all day because I'm very interested in that. but we are able to discover tens of thousands of species now that we previously weren't, especially microbial species, because of our use of environmental DNA and other extraction techniques. So the tree of life is growing more and more every day as we continue to do more research in this field, and we can see how interconnected things are, their common ancestors, and just where everything came from.
That's another topic that I didn't really get into, where this quantum biology may or may not be important, was DNA. But another time.
Yes, another time. But for now, thank you very much for joining us and listening to us, and have a good day. Bye, everyone.
Take care.
See ya.